Hi, I am Dr. Sean Patrick from the School of Health Systems and Public Health, and I'll be talking to you a little bit about the steps in research, and I will be giving you a practical example of some of my own work. For the purposes of this presentation, we will look at the way you move from a question to an answer. We will look at how you actually ask the question, look at designing the study, how you would collect the data on paper, how you would capture the data in electronic format, how you would take this data into a Stata program. Once you have analyzed in Stata, the results you get from there, and when you move into publishing the results. So from personal experience, I will talk to you a bit about the work that I did for my PhD. We had a look at what are the effects of environmental chemicals on the reproductive system. And while there were quite a large number of questions, the one question that, for the purpose of this presentation, we'll look at is, what is the impact of the in utero, lactational, and direct exposure of endocrine disruptive chemical exposure on male endocrine disruptive endpoints, as well as male accessory glands, and some of the other variables too. For the purposes of this presentation, I will focus on one of the male accessory glands, the prostate gland. What I will show you is why we chose the prostate gland, how the data was collected, how the data was inputted into Stata, what results we got, and how we actually visualize this in a presentation format. So we will look at how we ask the question, the process we follow to get to the publication where we answer the question. Right, so while we were thinking of the question, we had to think of how are we going to look at a study design that will answer these questions. So a study design that we chose for this study was an experimental study design. It involved a one-generation toxicological exposure study where we had one control group and 3 experimental groups. Now it sounds very complicated, but it's not that complicated. So let me explain to you. We started off with a certain number of pregnant females. They were split into a control group and 3 experimental groups. They were dosed daily through the duration of the pregnancy. Once the pups were born, they lactated and the pups were indirectly exposed through lactation for 3 weeks. Then the pups were directly exposed. For the purposes of this study, we only chose males, and the males were then split again into the control group into the 3 different experimental groups. These male pups were then exposed for 13 weeks, and at the end we terminated and we sampled. So at that point in time, we would then have a look at some of the variables, and one would include the prostate weight. So in order to answer my question, I had to control for the exposure, and I had to have 3 different experimental groups so that I could see, do any of these chemicals in these groups potentially have an effect on the prostate weight. So lab, and after the animals were dosed, we received the animals. I terminated the animals and I took out the prostate gland amongst the other glands, and they were weighed, and all the weights were recorded on paper first because we were in the lab and it was a bit crazy, but we recorded all the information on paper. Thereafter, it was my task to take all that information and put that into an electronic format. Here I included the group number as an identifier and the other different variables, and including the prostate weight over here. It's very important when you are transcribing from paper format to electronic format that you keep in mind to label your columns correctly and to have your data put in correctly too. The reason for that is when you take this information and you put this into any other statistical program that the data is captured correctly, that you get the correct information. So please make sure to constantly double check that you have the information that's on paper is correctly transferred to a electronic format. Thereafter, we took the electronic format in Excel, and that was then exported into Stata. Here in Stata, you would see the data. There's the group number, there's the other variables, and over here we have the prostate weights in the order that they were in my electronic format. It is also worthy to note that the numbers or the labels that you see on the top, this will be the very same labels that will be used once the statistical analysis has been done in order to compare the different groups. And here you can see the name of the group and what that label would mean. So we have prostate in grams, and as a file name, or as a column name rather, it was abbreviated to prostate G, so we could then see a difference between the two when we are comparing group 1 to group 2, etc. So I'm sure you would appreciate that because the study design was a little bit involved. We had pregnant females that were exposed, the pups were born, we then took the male pups and they were also then exposed. The reason that we did not use Excel to analyze the data was that we decided that if you have one female and she has many pups, that the differences you see in the pups might actually be related to the same female. So we needed to use a specific statistical test in order to look if they are, to determine if there are any confounding factors. So with the approach that we used was the survey. And in a survey, if you're surveying households, you have different households, so any differences you see could potentially be because of the different households. However, there are people living inside the household. So you could then say that people living inside one particular household, they might have the differences that might be attributed to the house. So if we translate that to this, the study that I'm talking about now, we could say that because many pups belong to one mother, the difference seen amongst the pups could potentially be attributed to the mother, and we needed to make sure that we corrected for that too. And for that reason, we used Stata. You will find when you going in depth into Stata that you will be able to analyze this, and it would make a bit more sense to you. So once information, we had run the survey command in Stata and looking particularly at the prostate gland. Here you would see the printout once the analysis has been done. There would be 16 females or 16 households. If you think back to the example that we had, the number of males was 77, that's the population size, and the printout, you would see there's group 1, 2, and 3. Those are your 3 experimental groups. What we are saying is we are comparing experimental group 1 to the control group, and if there is a significant difference between the two, we would look at the p-value, and the p-value is less than 0.05, which means yes, there is a significant difference between the prostate size in experimental group 1 and the control group. Now that we have this information, we'll look on the next slide how we translated this into a publication format. Here you see a table. This is a table that was used in the publication format. Group B is experimental group 1, group C is experimental group 2, and group D is experimental group 3, and our control group is group A. If you look at the prostate weight under mass here, you see that by comparing group B to group A, there is an increase in the prostate weight, and there is a significant difference there that is denoted by the p-value, the same p-value that we saw on the previous slide in the statistical analyses. So what I've shown you very briefly is we started off with a question. How does certain exposure to chemicals potentially have an impact on male reproductive health if we're using the prostate gland as a potential marker? We exposed the animals, we collected the data, we analyzed the data, and now in a publication format, we can show that yes, indeed, after doing the experiment, this is where we're reporting the data. Therefore, it is very important that your research question will ultimately determine the type of research study design that you use. So please, when you are looking at your own research questions, just think for yourself, how will I be able to answer this question? And later on, you will learn how to choose a different study design that will match up to your question. So I want to thank you very much for your time, and I wish you all the best for the rest of your module. Thank you.